Hello, everyone. Welcome to today's broadcast, Accelerating Method Transfer, Effective Automation of Biological Assays. I'm Rita Peters, Editorial Director of Pharmaceutical Technology and the moderator for today's event. We're pleased to bring you this educational web seminar presented by Pharmaceutical Technology and sponsored by Catalent. Catalent is the leading global provider of advanced delivery technologies and development solutions for drugs, biologics, and consumer health products. Catalent has over 80 years' experience in helping pharmaceutical, biotech, and consumer health innovators develop and supply superior products that improve people's lives. Catalent Biologics enables biologic therapies to get to patients faster through a suite of proprietary technology platforms and a range of integrated development, manufacturing, and analytical services. Catalent Biologics Analytical Services offer one of the broadest ranges of biologics analytical services in the industry, all under GLP and full GMP compliance. Catalent provides development services for drug substance, drug product, and intermediates. To find out more, please visit them on the web at www.catalent.com. Through cutting-edge science and medicine, Biogen discovers, develops, and delivers innovative therapies worldwide for people living with serious neurological and neurodegenerative diseases. Founded in 1978, Biogen is a pioneer in biotechnology, and today the company has the leading portfolio of medicines to treat multiple sclerosis, has introduced the first and only approved treatment for spinal muscular atrophy, and is at the forefront of neurology research for conditions including Alzheimer's disease, Parkinson's disease, and amyotrophic lateral sclerosis. Biogen also manufactures and commercializes biosimilars of advanced biologics. To learn more about them, please visit them on the web at www.biogen.com. I have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit your question by typing it in the Q&A box, which can be found on the right-hand side of your screen. You can enlarge the slide window by clicking on the small green icon in the upper right-hand corner of the slide window, or by hovering your mouse over the lower right-hand corner and dragging the window to the desired size. The slides will advance automatically during the event. And should you have any technical problems viewing or hearing the presentation, you can click on the question mark Help widget in the dock at the bottom of your presentation window. Before I introduce today's speakers, I'd like to get our audience involved in two brief polling questions. You can submit your answer by clicking directly on the screen. So here's the first question. Does your laboratory use automation for bioassay liquid handling? Yes, we have some automated assays. Yes, we have extensive automation. Yes, but we are just starting out. No, but it is planned within the next year. Or we have no current plans. So again, the question is, does your laboratory use automation for bioassay liquid handling? Yes, we have some automated assays. Yes, we have extensive automation. Yes, but we're just starting out. No, but is planned within the next year. Or we have no current plans. So you can make your selection on the screen and click Submit. And thank you for taking part in that poll. Let's move on to our second one. Once again, click directly on your screen to submit your answer. And the question is, where can you use some extra knowledge? And you can check all that apply. So for bioassay thaw-and-go cell banks, selecting the best mechanism of action-based bioassay development approach, bioassay lifecycle management for early to late-stage product release, increasing bioassay performance such as accuracy or repeatability, or analyst training and good documentation. Again, the question, where can you use some extra knowledge? With bioassay thaw-and-go cell banks, selecting the best mechanism of action-based bioassay development approach, bioassay lifecycle management, increasing bioassay performance, or analyst training and good documentation. So make your selection and click Submit. And thank you for participating in that. Let's move on to our program. I'd like to introduce today's three speakers. Len Blackwell, a principal scientist in the Analytical Development Automation Group at Biogen, leads efforts to improve bioprocesses at Biogen by delivering automated solutions with robotics and data analysis. He has 30 years of experience in the life sciences, spanning academic research, biotech, and pharmaceuticals. Len is passionate about the realization of complete automation in the biotechnology space. Christopher Hepler, Group Leader for Cellular and Molecular Technologies at Catalent Pharma Solutions, is a 20-year veteran of the pharmaceutical and biopharmaceutical industry. He has extensive validation and project management experience operating under good laboratory practices and good manufacturing practices. He has expertise in protein purification, biochemical assays, and laboratory automation. Tammy Conley, lead scientist for cellular and molecular technologies at Catalent Pharma Solutions, has more than 18 years of bioassay experience with an emphasis on cell-based potency assays. Her experience ranges from method development and optimization through validation, release, and stability testing. She manages a team of analysts and directs multiple projects. So thank you all for being here today. Tammy, I'm going to turn the presentation over to you.
Thank you, Rita, and thank you to the audience for joining us today for our webinar titled Accelerating Method Transfer, Effective Automation of Biological Assays. Today we'll talk about several key learning objectives. First, we'll talk about the advantages of automating your lab. These include improvements to safety and efficiency, throughput, and results that are accurate, reliable, and precise. Secondly, we'll talk about automation of biological assay processes. This includes an overview of a workstation from Hamilton, Catalent and Biogen's preferred workstation vendor. Third, we'll talk about a comparison of manual method transfer and automated method transfer, noting that robotic method performance is expected to be as good or better than manual method performance by removing the element of human variability. Next, we'll talk about instituting processes for successfully transferring automated methods. This is achieved by focusing on dedicated automation staff, effective communication between originating and receiving labs, and establishing realistic timelines. Lastly, we'll talk about working with automation-ready contract development organizations, or CDOs. In this section, we'll talk about what working with automation-ready strategic partners involves, including what considerations and services CDOs can provide to their partners. First, we'll talk about how automating your lab provides several advantages. Automation increases reproducibility through consistent performance gained by eliminating human-introduced variability seen between analysts and from day to day. The more reliable and reproducible your system is, the less rework is needed. Rework is costly and time-consuming. By improving your right first-time model, you are also able to greatly increase your efficiency. Automation allows analysts to multitask by reducing the touch time devoted to a single assay. Reducing analysts' touch time allows them to walk away to perform other high-value tasks. Automation reduces the need for hands-on skills within groups and dependency on skill coverage. This reduced dependence on experienced SMEs, subject matter experts, happens when you automate. Automation is not affected by skill or technique variations that occur between different analysts' pipetting abilities. The robot doesn't fatigue or get distracted. Automation allows skilled scientists to focus on the science and trust the automation to perform the more routine aspects of their work. Automation allows analysts to freely rotate among tests. Unlike analysts, automation doesn't require cross-training. Any automation-trained analyst can operate all of the program's protocols. Automation significantly reduces full-time employee, or FTE, time spent on a single task. Faster turnaround times and straightforward scheduling gives the analyst the ability to spend more time focusing on the results. Automation minimizes safety concerns by reducing ergonomic and biohazard risk. In our industry, carpal tunnel and other wrist and hand-related complaints related to repetitive motion, usually as a result of injuries incurred from pipetting, are a very real concern. Automation removes a great deal of that physical strain. Automation also reduces the risk of cross-contamination in multi-user environments, again, due to decreased touch time during assay execution. Another advantage of automation is the improved throughput seen by decreased turnaround time. Bioassays especially have a high FTE demand, are long and laborious procedures in most cases. Again, the instrument doesn't fatigue or need time off due to illness. Automation allows for increased sample numbers. We have the ability to miniaturize and move to smaller plate formats, such as a 96-well plate to a 384-well plate. We can fit more samples on a plate. We can fit more plates on a deck, and thus increase our throughput. Samples can be batched and processed all at once instead of running multiple times over a week. That allows the analyst to process their data one time a week instead of multiple times a week. And again, results variability due to analysts is eliminated, so investigation time and rework is reduced. Now I'll turn it over to Len Blackwell for the next section, Automation of Biological Assay Processes.
Thank you, Tammy. I'm going to discuss the automation of biological assay processes. One of the efforts that Biogen has put in automating biological processes is through the design of what we refer to as a bioassay robot. The robotics, as we've discussed earlier, involve the vendor, Hamilton, in particular their Microlab Star. We designed this instrument with custom integrations that are suitable to bioassay processes. For example, plate washing capabilities, a plate shaking ability, and plate reader for output. Also, within the instrument, we have within and off-deck plate moving capabilities to the integrated third-party instruments. The pipetting of the liquid is through both single-channel pipetters and a 96-well head. That gives us a lot of flexibility in our assay design. And lastly, we have an absorbance or a stop. Lastly, we have a barcode reader that can be employed through the process that can ensure that plates are placed in their proper positions, are recognized through the barcode and to what their identity is. As we had said, we use the Hamilton vendor for all of our robotics, and we've chose Hamilton because they have very unique technology, which makes the robots extremely reliable. In this in particular is how the instruments move liquid through their pipetting mechanisms. And this is referred to as their core technology, compressed O-ring expansion. This type of technology delivers the highest liquid handling precision as there's no vertical force or tip attachment or ejection. It eliminates mechanical stress on the instrument, which makes it more reliable. And it has the ability to pick up additional tools, for example, things other than tips, as in plate grippers. The Hamilton also has liquid level detection capability. This is a capacitive technology that senses the level of liquid, which is typically problematic in automating biological assays. This type of technology relates the robotic as closely as possible as the human hand. Thirdly, there is technology referred to as anti-droplet control. It just ensures that the pipetting mechanism is stable with highly volatile liquids, for example, with ethanol. And it prevents the pipette tips from leaking liquid. And then, as we referred to as into the safety of the instrument, it is an enclosed system. Its degree of enclosure can be custom designed. Fully enclosed to limit exposure is also HEPA compatible as well. And since it is enclosed, there's limited light exposure. So moving from the design of the robotics, this is one of our typical bioassay processes. The steps of the assay are noted below on the slide in blue and in yellow. And as we discussed before, bioassays in particular are extremely manual processes. And where automation delivers on these steps can be seen in this diagram, where a manual process, those initiated by the user, are shown in yellow among this 8-step process. And it's simply, in this case, setting up the robotic instrument and then taking the plate to its readout destination. All the steps in between, shown in blue, are carried out by the robot. And because of this, the analysts from the 2 yellow blocks give complete walkaway time where they're allowed to do other things. What I want to emphasize is, in addition to the robotics, there's a data process as well. These, as our laboratory processes, also can be labor-intensive, a movement of information and calculation of results. So we've designed complementary systems that automate the analytical processes in addition to the robotic methodology. Just as an example, this relies particularly on an electronic notebook, which I'll discuss later. But we can relate informatics that take place within a program, a robotic program where the robot creates information, analyzes it, and uses that information to carry out subsequent steps. Or we can move information to third-party software that does the calculations, places them in an electronic notebook, and then delivers it into our LIMS storage system. And the LIMS system, in our case at Biogen, is where our customers can submit samples and get their results. Going a little deeper into the electronic notebook, as you can see, it's the center of what we do. It is able to collate all the information that's required for an experimental test, including sample information, procedural information. It prepares a robotic work list. A work list is how a robot executes instructions that are related to a specific experiment. It is a house for raw data, and as I said before, it can do the analysis of the data itself. That workbook, in order to be useful, relies on the flow of information to other systems. In this case, it's fairly simple for illustrative purposes. We can move information from our LIMS system in an automated way to our workbook. In this case, it may be sample information, IDs, and preliminary results that factor into the calculation of a test. But we can also export results back into the LIMS system. Secondly, the workbook can then transfer information to our instruments, in this case in the robot. So if that's relating the sample information and execution of the assay to the robot, if there's a readout involved, that information then goes back into the workbook. So this is our ecosystem, as it were, of how we move information. So just to go one step further, again, in how we automate data analysis and report, I'd like to share with you the goals that we try to achieve when devising or designing this process. For one, we want to create a standard look and feel for any templates that we use for multiple platforms. And this is important when you deal with multiple analysts that run a variety of robotic methods. It's important that they have, in essence, a operating system that they can navigate through, and it looks the same for any test that they may carry out. The other goal is to simplify and consolidate information. Too often we find that our informatics sources rely either with an instrument or they're segregated in third-party software. We make the effort to bring those all together under one umbrella. Third is that we want to perform the analysis of raw data within the template so we don't rely on external analysis programs. Fourth, we want to automate the data analysis and flag any failed assays with acceptance criteria and employ automated data reduction. And this is important because as with assay execution in the laboratory, the same can be said for the analysis and interpretation of results. So if those are done in an automated way, we ensure that they're performed consistently. And lastly, we want to ensure that electronic workbooks align with other aspects of automation that we bring. For the last part of this, I'd like to share with you one of the workbooks that we use in our laboratory. This is a software package that is sold by IDBS. And in the first panel, or just on the far left, is just to give an idea of the flow of information so that we use the IDBS workbook with the circle. That information can be transferred to a work list to our robots, which then translate to plates that go to a reader, and that raw data then feeds back into the workbook. Shown on a table to the right of that diagram is what we call our launch pad. This is where analysts can navigate the aspects of a test, and it's broken down into a workflow going from samples in step 1 and 2 to results in the data analysis and reporting in number 5. It's from here that any analyst can navigate to particular tables depending on what the function that they're related to. So as I said earlier, this workbook contains tables of a variety of information, and I just want to share with you some of the key information that it's brought to in this workbook. And the first, through a data importing function, we bring in the raw data from the instrument. That raw data is then populated into a table where we have analysis of results. And as you can see in that analysis, we have in the green boxes where they say pass, we have acceptance criteria. And we have, where values are X'd out, we have the ability to have automated data reduction, which is essential for this particular bioassay. Next, we can look at the output of the information with a standard curve, and we can look at or achieve parameters for that curve so we can ensure that from assay to assay that's behaving as expected. And lastly, all the information is compiled into a summary where an analyst can review the results that have been reported, determine that criteria have or have not been met, and then select those results that can be passed on to our LIMS system and shared with our customers. That concludes the automation of biological assay processes. Next, I'm going to talk about the transfer of a manual method compared to an automated method. The automated method provides many advantages to the manual method, which I'm going to discuss. In both instances, these processes are, in essence, dependent on one another. And that from our approach, we use a data-driven process that relies on a statistical comparison of each process. It's within the first stage on the left where we have the originating lab. Essentially, we're dealing with a qualified method where we know essentially how the assay performs and a fairly good understanding of its variation. It's at this stage that we compare performance of a manual method and an automated method. Our goal at this stage is to ensure that the automated method can perform as good or better than a manual method. Whether a laboratory has automated processes or not, this stage, this comparison of manual and automated methods will probably happen under every circumstance. It is where the transfer, the movement of a method from an originating lab to a recipient lab where things can become advantageous. With a manual process, that means finding multiple analysts to run and train on the method so you ensure that its performance is comparable to the originating lab. This, as you can imagine, is very resource heavy in identifying those analysts and having backups and understanding the variability from analyst to analyst. With an automated method, it's merely a software transfer of method and ensuring that the performance is the same as the originating lab. And this, as I said, relies on the data itself and the statistics in comparing the two. Once that method has been operable, then it undergoes a performance verification in each case. And again, each verification is statistically driven, but in the case of the automated method, there's only one source and one robot, where in a manual process that involves many analysts to achieve the same result. Just to give a sense of what we look for when we compare, in this instance, automated and manual processes, this is information from a potency assay where we're looking at the recovery of a spike and whether that recovery floats around its expected value of 100% and then deviates from that based on the statistical variation of the assay. In this case, in the box on the right, you can see that the automated process shown in red is very comparable to the manual process shown with the black dots on the right. Both of these processes fit in the expected criteria, in this case of 80 and 125%. So from this information, we can reliably say that the automated method performs as good as the manual method. Where things get interesting is in the box on the left, where we look at the variation of the assay. Here, it's the ability of the data points to fit the predicted curve. And you can see that in the manual process, we see a higher degree of variance than the automated method. And in this instance, we can say that the automated method performs better than the manual process. Lastly, by achieving the transfer of an automated method and reliance on statistical information based on method performance, that gives us an extreme amount of versatility in transferring methods to either an internal quality organization, a CRO or CDO, or a global organization based on the same process. This is where automation has an extreme advantage over manual processes. Now I'm going to pass it on to Chris, who will look at instituting processes for successfully transferring automated methods.
Thank you, Len. To continue to build upon the theme of these processes, the next topic we would like to cover in this presentation is a discussion of having processes in place for method transfers. Having processes in place reduces the risk of missing steps and helps to ensure successful transfers that meet timelines. Catalent's process for successfully transferring automated methods is in an SOP. processes for method transfers are the same whether they are automated or not within that SOP. Key points of the SOP are communication, discussions of inputs and outputs, and the protocols and methods themselves. The first part of our process is having a kickoff meeting where members of the team from both the originating and receiving labs are introduced and discussions about expectations and timelines are discussed. Communication is the foundation of successful method transfer, whether they be automated or manual. The second part of our process is inputs and outputs. Inputs here refer to reagents and other consumables, steps, times, volumes, etc. Outputs would be data or results, any acceptance criteria or specifications that need to be discussed. One of the advantages for transferring automated methods is that we can focus on the data, as Len had mentioned previously, and not the specific manual training steps. An example of that would be not having to worry about if the analyst used the correct manual pipette or if they reversed pipette at a critical step. Those conversations can go away, and you can focus on data-driven decisions. The third part of our process is the protocol and the method. The goal is to allow an automated or manual process to deliver the same results. Let's expand on the assay method. The design of the automated method is identical to manual execution. You should have a single document that is not specific to the analyst or the instrumentation. The steps, times, volumes, and consumables of the assay are described, but it is understood that they are performed the same whether it is a robot or an analyst. A hybrid document may be required if there could be dependency on a manual execution, if the robot needed repair or another example. So manual automated methods may require hybrid documents where description of manual steps, note where steps are automated, and describe the operation of the robotics. The alternative method would be robotic focused, have a description of the robotic operation, describe the automated steps or scripts, and there would be no description of manual methods. So to have a complete process in place allows a successful tech transfer. So after the kickoff meeting and the writing the protocol and transferring the method, the script can then be pulled from a secure location, such as a cloud or a SharePoint site, and can be downloaded directly onto the workstation of the receiving lab. The method is then executed as it would be in a manual sense. The automated method is then executed with multiple runs over multiple days and the data compiled and statistically analyzed. In terms of the method transfer, the assay is validated before the transfer from the originating lab, and then the system at the new lab or the workstation at the new lab is qualified to the same acceptance criteria of multiple runs over multiple days with statistical analysis and predetermined acceptance criteria. Once they have been met and a method transfer report is written to capture these deliverables, the report is then signed off by both the originating and receiving laboratories, and the process is complete. The final topic of this presentation is a discussion about working with automation-ready CDMOs, that being a contract development manufacturing organization. What does a contract-ready CDMO look like, and what are some of the important points that a CDMO has? An automation-ready CDMO has three important parts that we'd like to discuss on the webinar today. The ability to work with automation-ready originating labs, having workstations in place, and having platform methods already scripted on workstations. To follow up on these three, partner installation, it's important for the CDMO to be flexible. Different partners will have different forms of automation. Is the automation like for like? Or will you have to do method writing? Will your strategic partner that you're working with want to be on site to do training, to do the method transfers? How involved will your partner want to be with the automation? You also may have partners that are virtual labs and that will rely on you to do method transfers from other locations and training of your staff and so on and so forth. So being flexible to partner installation is very important. The second important key part for automation-ready CDMO are the workstations. The first question to ask is if they're qualified and Part 11 compliant. The other question is there any redundancy? Do they have calibration and maintenance schedules that are important for compliance and other GMP questions? Finally, platform methods. Does the CMO have any generic platform methods that are already written for some assays? An example of that would be a host cell protein ELISA that is already a written method on the automation or on the workstation. And a second example would be PCR. Do they have analysts that are trained and an automation staff to support those activities? I would like to follow up on the validation, and this is an important piece of that picture. Here is a diagram of the validation process. Validation of instrumentation begins with initiating a change control and having a written validation plan. Once the validation plan is written, regulatory requirements and data integrity are important to the procedure. The user then writes requirement specifications to make sure that the instrument purchased and being qualified meets their requirements. The vendor usually comes in and does an installation and operational qualification. The laboratory will author the SOPs and then do some internal performance qualifications. When all of those have been successfully executed, there will be a validation summary report written, a change control will be closed, and the instrument will be released for use. All of these are very important steps in the validation process, and an automation-ready CDMO should have these procedures in place. This slide describes some of the necessary skills of an automation team. An automation-ready CDMO should have an automation team that has a direct line of customer service for the workstations that they have installed and qualified. They should also have informatics to allow for the automation of data and data analysis. Should also have information technology that will be very important to support the integration of various pieces of third-party equipment that may be integrated into the workstations. Another important aspect for an automation-ready CDMO is having project managers that have experience with workstations and automated methods and can interact with your customers with questions about the automation. Finally, the robotics and the decisions that were made in terms of purchasing the automation are also very important. At this point, I would like to turn the webinar over to Tammy.
Thank you, Chris. In summary, for today's webinar, we discussed advantages of using laboratory automation. We gave an overview of automation of biological assays. We showed a comparison of automated and manual method transfers. And we talked about the institution of processes to successfully transfer automated methods. And lastly, we talked about working with automation-ready CDOs. This concludes our presentation. Back to you, Rita.
Well, thank you, Tammy, Chris, and Len for the informative presentations. Before we get started on the Q&A, just want to remind our audience they can submit their questions by typing them in the Q&A box, which can be found on the right-hand side of the presentation window. And we'll try to answer as many questions as we can. So here's our first question. So how do I decide which liquid handler to purchase? Tammy, you have some insight on that?
Sure. So I can tell you what we did here at Catalent. We put together a team of people to discuss what sort of requirements and needs we thought we would have. We reached out to several vendors, and we arranged to visit their sites to see their instruments in action. Some of the sites that we visited were PerkinElmer, Hamilton, and TCAN. Len, Is there anything you'd like to add?
Yes, another great way to see instruments in action is to attend a vendor trade show. For example, there's a large vendor show that takes place once a year, the SLAS Conference, Society for Laboratory Robotics and Screening. And that's a great opportunity to see a vast array of robotics and devices and instruments. So I'd recommend anybody who were interested in seeing robots in action to attend one of these conferences. Also, in addition to the purchase itself, it's important to ask the vendor what type of support they provide after the purchase. And this can be training of personnel who are not familiar with robotics. This could be the types of service contracts they provide, or the accessibility to app support personnel or engineers. Is these types of features that really enhance the reliability and dependability on a particular vendor. Also, when looking at instrumentation, I'll consider the simplicity of the instrument itself. For example, if you're looking at air displacement, which is very similar to a pipette, for example, versus more complicated mechanisms. Also, you might look at efficient mechanical execution, and this ensures that you have fewer repairs with the instruments themselves.
Okay, thank you. Here's our second question. So how do I train users? Does the vendor take the lead?
Hello, Rita. This is Chris. Training is often part of the purchase package. In our case, Hamilton provided on-site vendor training, but they also offered an expensive off-site training, which we could have purchased and sent folks to. Len, do you have any follow-up?
Yes, one thing I think is important in a dependable automation program is to have in an institution a super user, one or two, that have very extensive knowledge into automation. And this can take place from training to the vendor itself, but is also perfected in the practice of automating procedures themselves. Once a super user has been designated, that person is relied upon to train, to develop methods to some degree, or work with the vendor to develop those methods, and then train personnel on those methods, and both running the methods themselves and to provide routine maintenance of the robots.
Okay, thank you, Len. Here's another question. Are service contracts useful? Tammy, what's your insight?
I personally think that service contracts are very important, and we feel that way as a company here at Catalent. If you do not have a fully staffed automation department, it really is a must. Catalent has a preventative maintenance schedule that's on a semi-annual basis. Len?
Yeah, I agree with Tammy. Service contracts are very important, and they typically ensure immediate service when you have a breakdown of the robots, which hopefully doesn't happen very often, but they do happen. And typically when you have these breakages, they tend to be things that are too complex for your super user, for example, or any internal resources. So having a service contract can be an effective part of a robotic program, especially if a site has no built-in redundancy. So in other words, they have no other robots to rely on while the operating robot is undergoing repair.
Okay, thank you. So is the use of an outside vendor to confirm precision of the liquid handler recommended?
From Catalent's perspective, that really should be addressed at a company level. For outside vendors can be cost prohibitive and expensive. What Catalent does is we rely on Hamilton to do that. They use a gravimetric method, and we do that at certain intervals, so we assure that the instruments are accurate and precise. Len, any comments?
Yes, we use typically other means to ensure the precision of a liquid handler. We've, for example, have designed specific processes that test every aspect of that through a gravimetric analysis, but we also use dyes and fluorescent compounds as well to determine precision.
Okay, thank you. Here's our next question. Is automation optional in a lab?
Catalent feels that once automation is implemented in the lab, only senior management or project directors should have the authority to revert to the use of the manual method, and only under special circumstances. Len?
Yes, automation processes, they're successful only if they're used. Likewise, the challenge of an automated process is that it's ensured to be efficient and adds value. Thus, an automation or an automated process undergoes a cycle of continuous improvements to ensure it is constantly improving and providing increased value to those who use it.
Thank you. Next question. How important is it to have workstation redundancy?
Workstation redundancy is important, especially if you're focusing on automated methods. It's important to have a second or third instrument that you can go to if your primary instrument or workstation is down for repairs. The other key part of that answer would be that was the discussion around having hybrid methods. So if you're going to write a hybrid method to get to your output either manually or from an automated fashion, it's important that you built that into your method as well. Len?
Yeah, I would say the necessity of redundancy is directly proportional to the dependency on automated processes. Just as an example, if the robotics are used weekly for multiple processes, having redundancy is going to be essential in maintaining timelines or turnaround times of sample results. However, if a timeline is more flexible, the wait time for repairs may be better tolerated.
Okay, thank you. Here's our next question. How are automated steps described and incorporated into an SOP?
It's important to write SOPs in a way that the method can be performed either manually or by automation, as we had discussed. For example, volumes, incubations, wash steps should be harmonized so that the methods can be performed manually or automatically. Steps within a method that can be automated can be emphasized with a footnote or an asterisk or something similar. Len, do you have anything to add?
Yeah, sure, Tammy. So when applying automation, one thing that's important is that the result isn't assigned and automated or assigned to be automated or manuals. And it's essential that the result of the automated and manual processes is within each method's variation. A given automated process should perform equally or better than the manual processes, as we said earlier. Thus, it's important that the automated and manual steps reside within a single SOP and not in an automated or manual SOP, respectively, for the same test. Additionally, instructions of automated steps, setting up the robotic deck, and navigating the software should reside independent of the manual steps, for example, in a job aid or in a segregated portion of the SOP, for example, a section or an indices.
Thank you, Len, and thank you, Tammy and Chris, for your presentations and for taking our audience questions. I'd like to thank our audience for joining us today, and I'd like to thank our sponsor, Catalent, for making today's webcast possible. Today's program will be available for on-demand viewing through November of 2018. You'll receive an email from Pharmaceutical Technology alerting you when this webcast will be available for replay. I invite you to forward that announcement to any colleagues who may have missed today's live event. So thank you for joining us, and we'll see you next time.